Welcome back to the Deep Dive, where we unpack your most complex sources and transform them into crystal clear understanding.
Hello again.
Today, we're diving into the fascinating and I think often overlooked world of sugars beyond just glucose.
Right. Glucose gets all the headlines, doesn't it? It's the body's primary energy currency, the main player everyone thinks about.
Exactly. But what about those other sugars we consume daily? The ones that might fly under the radar but play surprisingly crucial roles in our energy, our health, and even in how certain diseases develop.
We're talking about fructose and galactose.
Yeah.
These two monosaccharides may be less famous, but absolutely essential.
It's essential how?
Well, they contribute significantly to our energy supply, sure, but they're also key components of critical biological structures all through the body. So yeah, equally important players in your internal biochemistry.
Okay, so our mission for this deep dive is to really explore the intricate pathways of monosaccharide and disaccharide metabolism, zeroing in specifically on fructose and galactose.
Where they come from, how your body processes them differently, and it is different than glucose.
And crucially, what happens when those intricate pathways maybe hit a snag or go awry?
Exactly. Think of it as peeling back the layers of biochemistry, getting into the sweet science of your body, you could say.
And our sources for this are really solid, a comprehensive biochemical text. It gives us an incredible level of detail and insight, really letting us get to grips with this information, almost like the elite professionals in the field.
Yeah, it's a chance to see how really subtle differences at the molecular level can have these profound cascading effects on your entire physiological well-being. It's quite amazing, actually.
By the end, you'll definitely have a new appreciation for the precision of your body's internal machinery. Get ready to understand your diet and your body on a whole new level.
Let's dive in.
All right, let's start with fructose. When you think sugar, glucose might pop up first, but fructose is, well, it's an enormous player in our modern diet, often without us even realizing just how much.
It absolutely is. Fructose accounts for roughly 10% of the daily calories in the typical Western diet.
10%. That sounds like a lot when you think about it being just one type of sugar besides glucose.
It really adds up. For many people, that translates to about 55 grams of fructose per day. That's a substantial contribution to your overall energy intake, whether you're tracking it or not.
55 grams. Wow. Okay, so where is all this fructose coming from? I know it's in fruit, which we think of as healthy, but that can't be the whole story, can it? 55 grams feels like more than just an apple or two.
Oh, far from it. Yes, fructose is naturally found as a free monosaccharide in many fruits, and in honey, those are whole food sources, absolutely. But a major source, arguably the most significant in many diets, is actually the disaccharide sucrose.
Sucrose. So table sugar.
Exactly, common table sugar. The stuff you put in your coffee, what's in baked goods. When you eat sucrose, your intestine has enzymes that swiftly cleaved into two simpler sugars, equimolar amounts of fructose and glucose.
Equimolar, so half and half.
Pretty much exactly half and half. So essentially, half of the table sugar you consume is immediately turned into fructose right there in your gut.
Wow. Okay, so every time I add a spoonful of sugar to my tea, I'm basically adding half a spoonful of fructose. That definitely changes how I think about table sugar.
It adds a new dimension, doesn't it? And then, of course, there's the big one in processed foods.
Uh-huh.
High fructose corn syrup, HFCS.
Ah, yes. HFCS, that's everywhere.
It's ubiquitous, especially prominent in soft drinks, candies, baked goods, sauces, countless processed foods. While the exact mix can vary a bit, HFCS is typically composed of about 55% fructose and 45% glucose.
So even more fructose than glucose in that case.
Often, yes. Our source text specifically highlights its significant importance in processed foods, underscoring just how widespread this form of fructose is in our contemporary diet. Its prevalence is a major driver of that 10% daily caloric intake figure we mentioned earlier.
Okay, so fructose is truly everywhere. Table sugar, HFCS, fruit, honey. But here's where it gets really interesting, and a key difference from glucose: how it gets into our cells. My understanding is that it's not insulin dependent, unlike glucose in many tissues. Can you explain why that's such a critical point?
This is absolutely vital, and it sets fructose on a very different metabolic path right from the start. When you consume glucose, your pancreas releases insulin.
Right, the hormonal signal.
Exactly. And insulin acts like a key, unlocking the doors of many cells, think muscle cells, fat cells, allowing glucose to enter and be used for energy or stored. It's a tightly regulated process.
Like a traffic controller, directing glucose where it needs to go based on the body's needs.
Precisely. But fructose, it's a completely different story. Fructose transport into cells does not depend on insulin.
So it doesn't need the key?
It doesn't need the insulin key for entry into many key metabolic tissues like the liver. You can almost imagine fructose as having a VIP pass, bypassing that main insulin checkpoint. And furthermore, equally important, fructose itself does not stimulate the secretion of insulin from the pancreas.
Oh, wow. So it doesn't trigger its own regulation signal either.
Correct. This insulin independence means fructose can flood into cells, particularly liver cells, and be metabolized without the usual hormonal oversight that governs glucose. This lack of a controlled, insulin-mediated response has profound implications, especially considering the sheer volume we often consume. It lets fructose take a kind of fast track into certain metabolic pathways.
A fast track with no traffic lights. That sounds potentially problematic. Okay, so fructose gets this VIP pass into a cell, especially the liver. What's the very first biochemical step? What has to happen to it?
Well, like most sugars entering metabolism, it needs to be phosphorylated.
Right. Gotta add that phosphate group.
Exactly. Think of phosphorylation as sticking a chemical tag of phosphate group onto the sugar molecule. This tag does two crucial things. First, it effectively traps the sugar inside the cell. Phosphorylated sugars can't easily cross back out through the membrane.
Okay, traps it in. And second,
It chemically activates the sugar, essentially priming it, getting it ready for the subsequent enzymatic reactions down the line. It raises its energy level, making it more reactive.
and here for fructose phosphorylation, we have a bit of a tale of two enzymes, right? There's hexokinase, the general sugar phosphorylator, but also a specialist, fructokinase. How do they compare?
That's right. In most cells of your body, hexokinase is the workhorse enzyme.
Right.
It's responsible for phosphorylating glucose, converting it into glucose 6-phosphate. That's the classic first step of glycolysis.
The main pathway.
The main pathway. Now, hexokinase can technically phosphorylate other excesses, including fructose, but it's not very good at it.
Not very good. What does that mean in enzyme terms?
It means hexokinase has a high Michaelis constant, or kram, for fructose. Remember, kram is roughly an indicator of affinity, how well the enzyme binds its substrate. A high kram means low affinity.
So it doesn't grab onto fructose very tightly.
Exactly. Imagine hexokinase as a busy worker primarily focused on glucose. Even if fructose is around, hexokinase doesn't really see it or bind it effectively unless fructose concentrations are incredibly high. And under normal conditions, there's usually plenty of glucose around, which hexokinase much prefers. Glucose basically outcompetes fructose for hexokinase's attention.
So hexokinase is preoccupied with glucose, leaving fructose needing another way in, and that's where fructokinase steps up.
Precisely. Fructokinase is the enzyme that provides the primary and much more efficient mechanism for fructose phosphorylation. In stark contrast to hexokinase, fructokinase has a low matter for fructose, meaning high affinity. It binds fructose readily, and a high Vmax, or maximal velocity.
High Vmax means it works fast once it binds.
Very fast. It indicates exceptional efficiency and speed in handling fructose, even when fructose concentrations aren't super high. It's truly specialized for fructose, like a dedicated express lane just for it.
A fructose express lane. I like that. And where in the body do we find this express lane enzyme, fructokinase? Is it everywhere?
No, and its location is a key part of the story. Fructokinase is primarily found in the liver, the kidneys, and the small intestine.
Ah, the key organs that handle dietary absorption and processing.
Exactly. It rapidly converts fructose into fructose 1-phosphate, using ATP for that crucial phosphate tag. It's no coincidence that these three tissues are where most dietary fructose is absorbed and processed. And, importantly, these same tissues also contain a particular enzyme called aldolase B, which is critical for the very next step. This tissue specificity really highlights the body's targeted approach.
Okay, so fructose gets phosphorylated to fructose 1-phosphate in the liver, kidneys, small intestine. And this is where its path really diverges from glucose, right? Because glucose 6-phosphate goes on to become fructose 6-phosphate, then fructose 1-furan-6-bisphosphate in standard glycolysis.
Correct. Fructose 1-phosphate takes a different route. Unlike fructose 6-phosphate, which gets another phosphate added in glycolysis by phosphofructokinase 1.
The main regulatory step we mentioned.
Yes, that step. Fructose 1-phosphate bypasses that entirely. Instead, it's directly acted upon by aldolase B, sometimes called fructose 1-phosphate aldolase.
Aldolase B, what's its specific job here?
Aldolase B's job is to cleave fructose 1-phosphate directly into two smaller 3-carbon molecules. These are called trioses.
Trioses, 3 carbon sugars.
Exactly. The two products are dihydroxyacetone phosphate DHAP for short and glyceraldehyde. This cleavage step is absolutely critical because, as we hinted, it bypasses several key regulatory points in the main glycolysis pathway.
Okay, so it breaks fructose 1-phosphate into these two smaller pieces, DHAP and glyceraldehyde. You mentioned aldolase isozymes earlier. Are there other aldolases, and why is aldolase B the special one here?
Yes, the body actually makes three distinct aldolase isozymes, different versions of the enzyme from different genes. There's aldolase A, which is the most common one, found in most tissues playing a role in standard glycolysis. There's aldolase C, mainly found in the brain. And then there's our specialist, aldolase B.
And aldolase B is specifically in the liver, kidneys, and small intestine, just like fructokinase.
Precisely. Now here's the crucial distinction. All three aldolases A, B, and C can cleave the standard glycolysis intermediate fructose 1,6-bisphosphate.
Okay, they all do that job.
Right. But only aldolase B has the unique ability to efficiently cleave fructose 1-phosphate. Aldolase A and C just can't handle it effectively. This makes aldolase B absolutely indispensable for processing dietary fructose. Without it, fructose 1-phosphate just builds up, which, as we'll see, causes major problems.
Got it. So aldolase B is the key that unlocks the rest of fructose metabolism. And once we have these two trioses, DHAP and glyceraldehyde, what happens to them? Where do they go?
Both DHAP and glyceraldehyde are quite versatile and can slot right into existing metabolic pathways. Dihydroxyacetone phosphate, DHAP, is particularly flexible. It's actually an intermediate in standard glycolysis itself.
Oh, right.
So DHAP can seamlessly enter the glycolytic pathway to be burned for energy, or if the liver needs to make glucose, like during fasting, it can be shunted into gluconeogenesis.
Okay, so DHAP is easily handled. What about the other one, glyceraldehyde?
Glyceraldehyde is also quite adaptable, needs one more step, usually phosphorylation to become glyceraldehyde 3-phosphate, another standard glycolytic intermediate. So it too can feed directly into energy production or gluconeogenesis, or be used in other pathways like fat synthesis.
This all leads back to that fast track idea. You mentioned fructose metabolism is generally faster than glucose. Why is that again?
Yes, the rate is generally more rapid, and it comes down precisely to that pathway we just described. Fructose 1-phosphate, DHAP plus glyceraldehyde.
That sequence.
That sequence effectively bypasses the main regulatory checkpoint in glycolysis.
Which is, remind me again?
Phosphofructokinase 1, or PFK1. This enzyme catalyzes what's widely considered the major rate-limiting step in glycolysis. It's heavily regulated by the cell's energy status, hormones like insulin and glucagon. It's like a major, carefully controlled toll booth on the glucose highway.
Okay, a regulated toll booth.
But fructose, by being cleaved directly into DHAP and glyceraldehyde by aldolase B, essentially takes an unregulated side road that merges back onto the highway after that main toll booth.
Ah, it skips the traffic jam.
It skips the main regulatory control point. This means its metabolic flux, the rate at which it flows through these initial steps, is significantly faster and much less controlled compared to glucose, especially when consumed in large amounts.
And that rapid, less controlled flux, particularly in the liver, that's where potential issues can arise, like overwhelming the system.
That's a major part of the concern, yes.
Right.
Especially regarding conversion into intermediates that fuel fat synthesis, like triglycerides. The liver can get overwhelmed by a large fructose load.
Okay, so fructose is quickly metabolized, bypasses key controls. But what happens when these pathways themselves break down? What are the disorders of fructose metabolism? You mentioned they range from benign to severe.
Right. Deficiencies in these key enzymes can lead to distinct conditions.
Let's start with the more benign one you mentioned.
That would be essential fructosuria. This is caused by a deficiency in the first enzyme, fructokinase.
The one that does the initial phosphorylation.
Exactly. If fructokinase is deficient, fructose isn't efficiently phosphorylated to fructose 1-phosphate upon entering the cell. It can't be trapped or activated properly.
So what happens to the fructose?
It mostly just accumulates in the blood because it can't be processed efficiently, and then it spills over into the urine. That's why it's called fructosuria, fructose in the urine. It's generally considered benign because the fructose isn't getting trapped inside cells as fructose 1-phosphate and causing downstream havoc. It just kind of passes through.
Okay, so inconvenient maybe, but not dangerous. But then there's the other one, hereditary fructose intolerance, HFI. That sounds much more serious.
It is, yes. Significantly more serious. HFI occurs in about 1 out of every 20,000 live births. It's caused by a severe deficiency in the second key enzyme we discussed, aldolase B.
The enzyme specific to liver, kidney, and intestine that cleaves fructose 1-phosphate.
That's the one. And the symptoms of HFI are critical here. They don't usually appear right at birth. They typically start when a baby is weaned from breast milk or formula that's primarily lactose-based.
Lactose being glucose and galactose. No fructose there.
Right. Symptoms appear when the baby starts ingesting foods containing sucrose, which breaks down to fructose and glucose, or free fructose itself. Things like Fruit juices, sweetened cereals, some fruits, sometimes even certain medications or formulas sweetened with sucrose or fructose.
And what happens then when that fructose hits their system and aldolase B isn't working? What's the biochemical cascade?
This is where it gets really problematic. Without functional aldolase B, fructose 1-phosphate, that product of the first step, starts to accumulate massively inside the cells, especially in the liver, kidneys, and small intestine.
It can't be broken down, so it just builds up.
It builds up rapidly. And this accumulation triggers a devastating cascade of biochemical consequences. It's like dominoes falling.
Okay, domino number one.
Domino number one. Fructose 1-phosphate acts as a potent phosphate trap. It ties up inorganic phosphate pi, which is absolutely vital for cellular energy. This causes a dramatic drop in the intracellular levels of free inorganic phosphate.
And why is dropping phosphate levels so bad?
Because inorganic phosphate is an essential ingredient for making ATP, the cell's main energy currency. It's literally part of the ATP molecule. If pi levels plummet, ATP production grinds to a halt.
No phosphate, no ATP.
Pretty much. The cell simply cannot generate enough ATP to power its countless metabolic reactions. As ATP levels fall, another molecule, adenosine monophosphate, or AMP, a breakdown product of ATP, starts to rise.
Okay.
The body tries to get rid of this excess AMP by rapidly degrading it. But this degradation pathway has byproducts. It causes a significant increase in uric acid, leading to hyperuricemia, too much uric acid in the blood, and it also contributes to lactic acidemia, too much lactic acid.
So not only is the cell starved for energy, low ATP, but it's also accumulating toxic byproducts like uric acid and lactic acid.
Exactly. It's a double whammy, putting immense strain on the liver and kidneys.
What other effects does this severe lack of ATP have, particularly in the liver?
The decreased hepatic ATP has widespread life-threatening effects. A huge one is that it cripples gluconeogenesis.
The liver's process for making new glucose.
Yes, the vital process for maintaining blood sugar levels, especially between meals or during fasting. When gluconeogenesis shuts down due to lack of ATP, the person experiences profound and dangerous hypoglycemia, low blood sugar, often accompanied by vomiting. The liver just can't keep blood glucose stable.
That sounds incredibly dangerous, especially for an infant.
It is. Additionally, the lack of ATP severely impairs protein synthesis. The liver needs ATP to make crucial proteins, including blood clotting factors. So you can see issues with bleeding. It also affects the synthesis of other essential proteins and enzymes.
Okay.
Our source also notes decreased kidney reabsorption of phosphate, making the phosphate depletion worse. And importantly, the drop in inorganic phosphate also powerfully inhibits glycogenolysis.
Glycogenolysis, breakdown of stored glycogen.
Yes. So the liver can't even efficiently break down its stored glucose, glycogen, to help with the hypoglycemia, because that process also requires inorganic phosphate. The body is essentially crippled on multiple fronts when it comes to producing, storing, and regulating its primary fuel, glucose.
That is a truly devastating chain reaction, all from one missing enzyme, aldolase B. How is HFI diagnosed? Can it be caught early?
Diagnosis can involve a few approaches. Clinicians might detect fructose in the urine, though less reliable than an essential fructosuria. A more definitive test is an enzyme assay using liver cells, perhaps from a biopsy, to directly measure aldolase B activity. But increasingly, and much less invasively, DNA-based testing is used to look for specific known mutations in the aldolase B gene.
And the treatment, given how severe it is.
The treatment is absolutely critical and, thankfully, very effective if started early and followed strictly. Complete and lifelong removal of all sources of sucrose and fructose from the diet.
Complete removal. That must be challenging.
It requires extremely careful dietary management and label reading forever, but it prevents that toxic accumulation of fructose 1-phosphate and the whole devastating biochemical cascade. It can prevent the severe liver failure, kidney damage, growth problems, and potential death associated with untreated HFI.
And there's that interesting quirk about people with HFI and sweets, right?
Yes, it's quite fascinating because ingesting even small amounts of fructose or sucrose causes such immediate and severe symptoms, nausea, vomiting, pain. Individuals with HFI typically develop a very strong natural aversion to sweet-tasting foods very early in life.
A built-in protective mechanism.
Exactly. They learn instinctively to avoid what makes them sick. And as a rather unique consequence of lifelong avoidance of dietary sugars known to cause cavities, they often have a remarkable absence of dental caries. A small silver lining, perhaps.
Okay, that covers fructose metabolism and its disorders in incredible detail. Now, briefly, what about mannose? It's another sugar, but not one we hear much about dietarily. How does it fit in?
Right. Mannose isn't a major dietary energy source for us. Its role is much more structural. Biochemically, mannose is the C2 epimer of glucose.
Epimer, meaning it's almost identical to glucose, but flipped at one carbon.
Exactly. Just the configuration around the second carbon atom is different. This subtle difference matters. Mannose is a really important component of glycoproteins.
Proteins with sugar chains attached.
Yes, complex molecules crucial for all sorts of things. Cell recognition, immune function, tissue structure. Mannose is a key building block for these sugar chains.
So if we don't eat much mannose, how does it get into our metabolic pathways or into these glycoproteins?
Primarily through internal pathways. Hexokinase, the same enzyme that phosphorylates glucose, can also phosphorylate mannose to mannose 6-phosphate.
Okay.
Then another enzyme, phosphomannose isomerase, reversibly converts mannose 6-phosphate into fructose 6-phosphate.
Ah, there's the link. It feeds into the fructose glycolysis pathway.
Exactly. It provides a way for mannose to enter mainstream metabolism. But most intracellular mannose isn't coming directly from the diet. It's either synthesized internally, often from fructose precursors, or it's salvaged and recycled when old glycoproteins are broken down by the cell. Dietary carbs have very little free mannose.
Got it. So more of an internal building block and recycling story. Now let's shift to another pathway, one that actually converts glucose into fructose via something called sorbitol. This sounds like one of those pathways with both good and bad sides.
It is indeed a classic biochemical double-edged sword. This is often called the polyol pathway. The basic idea is converting a monosaccharide, like glucose, into its corresponding polyol, or sugar alcohol.
Sugar alcohol, like sorbitol.
Exactly. Glucose gets converted to sorbitol, also called glucidol. This happens when the aldehyde group on the glucose molecule is reduced to an alcohol group adding a hydroxyl group.
Okay, why do that? What's the point of making sorbitol?
A key consequence, and sometimes the point, is that polyols like sorbitol are effectively trapped inside the cell where they're made. Unlike sugars or their phosphate esters, they don't easily cross cell membranes without specific transporters, which aren't always present.
So making sorbitol is a way to keep a glucose-derived molecule inside certain cells.
It can function that way, yes.
Yeah.
The enzyme that does this first step, converting glucose to sorbitol, is called aldose reductase.
Aldose reductase. What's important about it?
Two key things. First, it uses NADPH as a cofactor, which we'll come back to. Second, it has relatively high kilometer for glucose.
High kilometers again, meaning low affinity. It only works well when glucose is abundant.
Precisely. Under normal blood glucose levels, aldose reductase isn't super active. But when glucose levels are high, like in hyperglycaemia, this enzyme kicks into gear. It's found in many tissues, including some particularly vulnerable ones, the retina, the lens of the eye, kidneys, peripheral nerves, ovaries, and seminal vesicles.
Okay, so aldose reductase makes sorbitol when glucose is high. Is that the end of the pathway?
No, there's a second step in some tissues. Another enzyme, sorbitol dehydrogenase, can then oxidize sorbitol into fructose.
Ah, so glucose to sorbitol fructose. Where does the second step happen?
Sorbitol dehydrogenase is found mainly in the liver, ovaries, and significantly the seminal vesicles.
Seminal vesicles, that sounds specific. Is there a physiological benefit there?
Yes, there seems to be. Sperm cells actually utilize fructose as a major energy source for motility. So this two-step pathway provides a neat way for the seminal vesicles to synthesize fructose directly from the glucose available in the bloodstream, providing fuel tailored for sperm.
Okay, that makes sense. And maybe in the liver, too, it just provides another route to get fuel into glycolysis.
Exactly. The liver can convert sorbitol, if present, into fructose, which can then easily enter glycolysis via fructokinase. So it has some normal physiological roles.
But you called it a double-edged sword. Where does the dark side come in? This sounds like the diabetes connection.
This is absolutely where the major pathological problems arise, particularly in uncontrolled diabetes. Remember, aldose reductase kicks in strongly during hyperglycemia.
Right, when blood glucose is high.
And critically, remember that glucose entry into cells of the retina, lens, kidneys, and peripheral nerves does not require insulin.
Ah, they are free entry zones for glucose regardless of insulin.
Exactly. So in someone with uncontrolled diabetes and high blood sugar, massive amounts of glucose just flood into these specific vulnerable cells. There's no insulin gatekeeper to limit the influx.
And with all that glucose inside.
And plenty of NADPH usually available, aldose reductase goes into overdrive, producing large amounts of sorbitol inside these cells.
Okay, lots of sorbitol being made. But you said it gets trapped?
It gets trapped.
Mm-hmm.
Sorbitol cannot easily pass back out through the cell membranes. And crucially, many of these same vulnerable tissues, the retina, lens, kidneys, peripheral nerves, have very low levels or are completely lacking the second enzyme, sorbitol dehydrogenase.
So they can make sorbitol from glucose, but they can't easily convert it to fructose or get rid of it.
Precisely. Glucose goes in, gets converted to sorbitol, and then the sorbitol just accumulates. It builds up inside the cell.
Like filling a room you can't easily empty. What does that accumulation do to the cell?
It causes major osmotic stress. Sorbitol is an osmotically active molecule. Like salt, it draws water towards it. As sorbitol piles up inside the cell, it pulls water in from the outside via osmosis.
Causing the cell to swell up.
Exactly. The cells swell due to water influx and retention. This chronic osmotic stress disrupts normal cellular function, damages delicate internal structures, messes up ion balances, and eventually can lead to cell injury and death.
And this osmotic stress, this swelling and damage caused by trapped sorbitol, this is directly linked to diabetic complications.
It's considered a major contributing factor to some of the most severe long-term complications of poorly controlled diabetes, such as cataract formation, the clouding of the lens due to swelling and protein changes, peripheral neuropathy, nerve damage causing pain, numbness, tingling, often in the feet and hands, and microvascular problems leading to nephropathy, kidney damage, potentially leading to kidney failure and retinopathy damage to the small blood vessels in the retina, which can cause vision loss and blindness.
Wow. Cataracts, neuropathy, kidney damage, blindness, all linked back to this seemingly simple pathway turning excess glucose into trapped sorbitol.
It's a powerful example of how a metabolic side pathway, when chronically overactivated by high glucose, can wreak havoc.
And there was something about NADPH, too, an antioxidant connection.
Yes, an additional insult, really. Remember, aldose reductase uses NADPH to convert glucose to sorbitol. Well, NADPH is also critically needed by another enzyme system to regenerate reduced glutathione. Glutathione is one of the body's most important antioxidants, protecting cells against damage from reactive oxygen species.
So making too much sorbitol uses up the NADPH needed for antioxidant defense.
Exactly. By diverting NADPH towards sorbitol production during hyperglycemia, the cell's ability to maintain its glutathione-based antioxidant shield is compromised. This likely leads to increased oxidative stress, further damaging these vulnerable cells and probably accelerating the progression of those diabetic complications. It's a truly insidious cycle. High glucose leads to sorbitol, which causes osmotic stress. ANE depletes antioxidant defenses.
That's incredibly illuminating. Understanding that detailed biochemistry really clarifies the mechanisms behind those devastating complications. Okay, let's shift gears now to our other main player, galactose. We mostly get this from milk, right?
Yes, that's the primary source for most people. The major dietary source of galactose is lactose, the disaccharide in milk and dairy products. Lactose is specifically structured as galactosyl Bast1-Vol4-glucose.
And we digest that in the intestine.
Correct. The enzyme lactase, which is technically galactosidase, sits on the surface of intestinal cells and cleaves lactose into its two monosaccharide components, one molecule of glucose and one molecule of galactose. These are then absorbed.
So milk gives us galactose. Any other sources?
We also get some galactose internally from the normal breakdown, the lysosomal degradation of the body's own complex molecules like glycoproteins and glycolipids, which contain galactose units. But dietary lactose is usually the main input.
And like fructose, galactose entry into cells is also insulin independent.
That's correct. Another parallel there. Galactose transport into cells doesn't require insulin.
Okay, so galactose gets into the cell. First step, same as the others. Phosphorylation.
You got it. Galactose must be phosphorylated to be trapped and metabolized. Most tissues have a specific enzyme for this, galactokinase. It uses ATP to add a phosphate group, producing galactose 1-phosphate.
Galactose 1-phosphate. Now, how does that get into the main energy pathways like glycolysis? Because it's not a direct intermediate, is it?
No, it's not. Galactose 1-phosphate cannot directly enter glycolysis. It needs to be converted into a form the main pathways recognize. Specifically, it needs to become UDP galactose.
UDP galactose. What's UDP?
UDP stands for uridine diphosphate. Think of UDP as another kind of chemical tag or handle, like the phosphate group, but this one is used specifically to carry sugars around for certain reactions, particularly biosynthetic ones.
Okay, so galactose 1-phosphate needs to become UDP galactose. How does that happen?
It happens through a really neat exchange reaction. A molecule called UDP glucose reacts with the galactose 1-phosphate.
UDP glucose. So glucose with that UDP tag.
Exactly. In the reaction, the galactose 1-phosphate essentially swaps its phosphate group for the UDP glucose's UDP tag. The result is UDP galactose and glucose 1-phosphate.
A direct swap. What enzyme does this?
This crucial exchange is catalyzed by galactose 1-phosphate, uridylyltransferase, mercifully abbreviated as GALT. This enzyme is absolutely key for integrating galactose into metabolism.
And the glucose 1-phosphate that's also produced.
That's easily handled. Glucose 1-phosphate can be readily isomerized to glucose 6-phosphate, which slots right into glycolysis or other glucose pathways.
Okay, so Galtech makes UDP galactose. Now, how does that fit into the bigger picture? Can it enter glycolysis directly?
Not quite directly as UDP galactose. For the galactose unit within UDP galactose to enter the mainstream of glucose metabolism, it usually needs one more transformation. It needs to be converted to its C4 epimer.
Another epimer switch, like glucose and mannose at C2.
Exactly. But this time at carbon 4. UDP galactose needs to be isomerized to UDP glucose. The enzyme that does this is UDP hexose 4 epimerase.
So it flips the configuration at carbon 4, turning UDP galactose into UDP glucose.
Precisely. And this new UDP glucose, originally derived from galactose, is now identical to UDP glucose made directly from glucose. It's incredibly versatile. It can be used in various biosynthetic reactions like making glycogen, or it can even participate back in that Galt reaction we just talked about, allowing the UDP tag to be recycled. It shows how interconnected these pathways are.
But UDP galactose itself, before being converted to UDP glucose, it has its own important roles, right? You mentioned biosynthesis.
Absolutely. UDP galactose is not just an intermediate on the way to energy. It serves as the direct donor of galactose units in a whole range of vital synthetic pathways,
Such as?
such as the synthesis of lactose itself, which we'll get to,
Yeah.
but also critically in building those complex carbohydrates we mentioned earlier, glycoproteins, glycolipids, and glycosaminoglycans. UDP galactose provides the galactose building blocks for these essential structural and signaling molecules found throughout the body in cell membranes, connective tissue, mucus, etc.
So it's a crucial building block.
Yeah.
Now, here's an interesting question. What if someone can't consume dietary galactose? Say they're lactose intolerant. Can their body still make these essential glycoproteins and glycolipids that need galactose?
That's a great question, and it highlights the body's amazing flexibility. The answer is yes, absolutely. If dietary galactose is absent, the body can still meet all its tissue requirements for UDP galactose.
Oh.
It uses that enzyme UDP hexose 4 epimerase, but in reverse. It takes readily available UDP glucose, which can be easily made from glucose, and converts it into UDP galactose.
So the epimerase reaction is reversible. It can go both ways.
Yes, it allows the body to synthesize galactose endogenously from glucose whenever it's needed for these crucial biosynthetic pathways. It's a fantastic backup system, ensuring we can always make these vital complex carbs, regardless of dietary galactose intake.
That's remarkable biochemical adaptability, but just like fructose, things can go wrong with galactose metabolism too, leading to disorders. What's the main one?
The most severe disorder is classic galactosemia. This is caused by the severe deficiency or complete absence of the GALT enzyme.
Jolts here, the enzyme that does the UDP swap, converting galactose 1-phosphate to UDP galactose.
That's the one. If GALT is missing or non-functional, galactose 1-phosphate cannot be converted.
So, similar to HFI, the intermediate builds up.
Exactly. Galactose 1-phosphate accumulates rapidly and dangerously inside cells, and consequently, free galactose also builds up in the blood and tissues.
What are the consequences of this buildup? Is it similar to HFI?
The physiological consequences are similar in their damaging nature to HFI, severe liver dysfunction, hypoglycemia potential, but classic galactosemia often affects a broader range of tissues more severely, especially early in life. Brain development is particularly vulnerable.
And does galactose get shunted into side pathways like fructose does?
Yes, it does. The excess galactose gets acted upon by aldose reductase, that same enzyme from the polyol pathway and diabetic complications.
Aldose reductase again, what does it do to galactose?
It reduces galactose to galactitol, which is the sugar alcohol of galactose, analogous to sorbitol from glucose.
And galactitol, like sorbitol, causes osmotic problems.
Precisely. Galactitol is osmotically active and poorly metabolized. Its accumulation, along with galactose 1-phosphate, contributes significantly to the damage. It causes major liver damage, often leading to jaundice, cirrhosis, and liver failure in infants if untreated. It causes severe brain damage, leading to intellectual disability and neurological problems. And it causes cataracts very rapidly, often visible within days or weeks of birth if the infant is consuming lactose.
That sounds absolutely devastating. How is it diagnosed and treated?
Thankfully, because it's so severe and requires immediate intervention, classic galactosemia is included in newborn screening panels in many countries. A blood spot test can detect low GALT activity or high galactose levels within days of birth.
So it can be caught very early?
Yes, and early diagnosis is critical. The treatment is, like HFI, dietary. It requires the complete and lifelong elimination of all galactose and lactose from the diet. For infants, this means specialized soy-based or elemental formulas that contain no lactose or galactose. For life, it means strict avoidance of all milk and dairy products and careful checking of labels for hidden sources.
And does that strict diet prevent the long-term damage?
If started immediately after birth, it can prevent the acute life-threatening liver failure and cataracts and significantly improve the long-term outcome. However, even with good dietary control, some individuals with classic galactosemia may still experience long-term complications like learning difficulties or ovarian issues in females, suggesting the underlying metabolic disruption might have effects beyond just the acute toxicity. It's a complex condition.
Are there less severe forms?
Yes, there are. Deficiencies in the other enzymes of the pathway galactokinase, the first step, or the UDP hexose-4 epimerase also cause disorders of galactose metabolism. These are generally less severe than classic GALT deficiency, likely because the toxic galactose 1-phosphate doesn't accumulate as dramatically, or there are alternative routes. However, cataracts are a common feature even in these milder forms, highlighting the lens's particular sensitivity to galactose and galactidal accumulation.
Okay, we've explored fructose and galactose metabolism individually. Now let's connect back to lactose itself. We know it's the source of dietary galactose. But how is this crucial milk sugar actually made by the body? This seems like a really elegant piece of biochemistry.
It truly is. It's a beautiful example of specific enzyme regulation for a very specific purpose. So lactose, as we said, is a disaccharide. One molecule of agalactose linked by a specific glycosidic bond to a molecule of glucose, lactosyl 1-kinase glucose.
And it's made only in the mammary gland during lactation.
Correct. It's synthesized de novo from scratch within the Golgi apparatus of the epithelial cells in lactating mammary glands. It's the defining sugar of mammalian milk.
How does the cell stick that galactose onto glucose? What's the enzyme?
The enzyme responsible is called lactosynthetase. Its more formal name is UDP galactose, galactosyltransferase, which describes exactly what it does. It transfers galactose from UDP galactose onto glucose, releasing the UDP tag in the process.
Lactosynthase. But here's the really cool part, right? This enzyme isn't just a single protein.
Exactly. This is what makes it so unique. Lactose synthase is actually a complex composed of two completely different proteins that have to come together to do this specific job. They're known simply as protein A and protein B.
Okay, protein A and protein B. Let's start with protein A. What is it, and is it only in the mammary gland?
Protein A is an enzyme called berry D-galactosyltransferase. And interestingly, no, it's not exclusive to the mammary gland. Protein A is actually found in many different tissues throughout the body.
Doing what in those other tissues? Not making lactose, presumably.
Correct. In tissues other than the lactating mammary gland, protein A uses UDP galactose, but it transfers the galactose onto a different acceptor molecule. Instead of glucose, it transfers galactose onto N-acetyl D-glucosamine.
N-acetyl D-glucosamine. What does that make?
It forms a disaccharide unit called N-acetyl lactosamine. This molecule is a fundamental building block of the complex carbohydrate chains found on many N-linked glycoproteins.
Ah, so protein A on its own is involved in making those structural glycoproteins we talked about earlier.
Precisely. It plays a general, important role in glycoprotein synthesis across the body, adding galactose units to build these complex structures. It's a versatile galactosyltransferase.
Okay, so protein A is the general galactose transferase. Then what about protein B? This must be the mammary gland specialist.
Exactly. Protein B is the key regulator here. It's a protein called lactalbumin. And crucially, lactalbumin is found exclusively in lactating mammary glands. Its synthesis is turned on specifically by the hormone prolactin.
Prolactin, the hormone that stimulates milk production itself.
Yes, prolactin levels rise significantly during late pregnancy and after childbirth, switching on the production of lactalbumin, protein B, only when milk needs to be made.
Okay, so we have protein A, the general transferase, always around, and protein B, lactalbumin, only there during lactation. How do they interact? How does protein B change what protein A does?
This is the elegant switch. When protein B, lactalbumin, is present, it binds to protein A, the galactosyltransferase, and forms a complex, the functional lactose synthase enzyme.
And what does forming this complex do?
It dramatically changes the substrate specificity of protein A. Specifically, the binding of lactalbumin causes a conformational change in protein A that drastically lowers its kilometers for glucose. Remember, low kilometer means high affinity.
So putting protein B onto protein A suddenly makes protein A grab onto glucose much, much more tightly, like thousands of times more tightly.
Exactly, by about 1,000-fold, in fact. On its own, protein A barely interacts with glucose, prefers N-acetylglucosamine. But when a lactalbumin binds, it essentially reshapes protein A's active site, making glucose its overwhelmingly preferred acceptor substrate. It's like lactalbumin acts as a modifier, telling protein A, okay, now focus on glucose.
That's incredible. So the presence of this one regulatory protein, lactalbumin, completely switches the enzyme's function from making glycoprotein components to mass-producing lactose.
Precisely. This crucial shift ensures that in the lactating mammary gland, where glucose is readily available, the enzyme complex churns out lactose, galactose plus glucose, in large quantities, rather than N-acetylactosamine, galactose plus N-acetylglucosamine. It's a brilliant biological switch, using a hormone-induced regulatory protein, B, to modify the activity of a common biosynthetic enzyme for a highly specific temporary physiological need, making milk sugar. Without this lactalbumin switch, milk wouldn't contain lactose.
This has truly been an incredible deep dive. We've gone through the metabolism of fructose and galactose, uncovering these unique paths from our diet right down to cellular function. It's really striking how different their processing is from glucose, especially that insulin independence and the specific enzymes involved, like fructokinase and aldolase B, or JOLT for galactose.
It really highlights the specificity. And what's truly striking, I think, is how seemingly minor enzymatic differences, like aldolase B being the only one that can cleave fructose 1-phosphate, or the dual nature of aldose reductase causing problems in both diabetes and galactosemia, can lead to such profound and widespread health consequences.
Yeah, from relatively benign conditions like essential fructosuria all the way to severe life-threatening disorders like HFI and classic galactosemia. It just underscores the body's incredible biochemical precision, doesn't it?
Absolutely. Every enzyme, every pathway exists in this delicate balance. When that balance is disturbed, even by one faulty enzyme, the ripple effects can be enormous, impacting energy levels, brain function, vision, liver health, everything. It shows there's so much more to sugar metabolism than just providing calories.
It really does. And this detailed understanding isn't just about rare genetic disorders, is it? It gives us critical insight into much more common health challenges, too.
Exactly. Think about our modern diet, which is often incredibly rich in added fructose, especially from sucrose and high fructose corn syrup. How does that massive influx interact with these ancient metabolic pathways, particularly the less regulated fast track in the liver? Understanding the details helps explain why excessive consumption of fructose might have unique detrimental effects on things like liver fat accumulation and metabolic syndrome, beyond just its calorie count.
It empowers us to look beyond simple calorie counting and appreciate the really intricate molecular dance that's happening inside us every single day based on what we eat. So considering everything we've uncovered today, what's a final provocative thought for our listeners to maybe chew on?
Well, it makes you wonder, doesn't it? We've focused on fructose and galactose, these other sugars. What other common dietary components, maybe specific types of fats, amino acids, or even micronutrients, might be influencing our biochemistry through similarly intricate specific pathways that we're only just beginning to fully understand at this molecular level? Yeah. The more we uncover these mechanisms, the more we realize the profound and often very specific impact our daily dietary choices have on that delicate, finely tuned biochemical balance within our bodies. It certainly prompts a deeper appreciation for the sweet and sometimes complex science of life itself.